In this video, we're going to take a look at Chapter 11, Section 3, the structure of complex carbohydrates. Complex carbohydrates are disaccharides, and this is an example here. Sucrose is table sugar, as we know it commonly, and it is a glycosidic bond length between the 6-carbon sugar glucose and the 5-carbon sugar fructose. Disaccharides yield 2 monosaccharides when hydrolyzed. Hydrolyzation will break this glycosidic bond here. So their definition, they're identified by their monosaccharide components. Monosaccharides are joined by this glycosidic bond between the anomeric carbon, which is right here, and the OH kicks off a hydrogen to allow for this glycosidic bond. This is a beta because it's pointing upwards. So, in contrast to the anomeric center of the monosaccharide, the anomeric center at the glycoside linkage is stable. So it does not open and close in aqueous solution. So remember, with our monosaccharides, we're practicing drawing with the Fischer projection and then back to the Haworth projection. That's in a link because remember, in solution we have some ringed sugars, some unringed, like the Fischer projection in a solution, and then some in between. So they're always in flux. But that's not so with the disaccharides. They're stable. They don't open, they close, and they're closed in solution, so they cannot change from alpha to the beta anomers. So once they are beta here, which the oxygen is pointing up from the anomeric carbon here in the alpha it's pointing down, they stay there. In order to release that glycosidic bond, we need a catalyst. So a catalyst is something it's in the environment. Often it's an enzyme protein in the body, or if it's in industry, it's a metal that lowers the activation energy, makes a reaction happen faster because it lowers the energy that the collisions need to happen between the molecules to break and form new bonds. So we're going to use acid to hydrolyze glycosidic bonds, and we're going to explore carbohydrates with our next lab next Tuesday. In biological systems, a specific enzyme that doesn't work elsewhere, it catalyzes the hydrolysis of glycosidic bonds. Lactose is the disaccharide found in milk, and it's hydrolyzed to D-galactose and D-glucose with the enzyme lactase. So remember, chemicals that end in the suffix ASER enzymes. So these 2 monosaccharides are joined by a beta, so the oxygen points up from the anomeric carbon of galactose, and it's a 1,4 linkage. So 1 on the galactose ring, 4 on the glucose ring. The second anomeric carbon is not stabilized and so can open and close. This one. So this glycosidic bond stays, but this glucose ring is opening and closing. Fructose is the most common disaccharide. That's table sugar. It's in many fruits. It's unique because the glycosidic linkage occurs between anomeric carbon centers of both, which is one of the reasons it doesn't open and close. So it's a 1-2 bond, which is unique. Both rings are stable and neither ring can open. Disaccharides that connect at both anomeric centers are known as non-reducing sugars, and that's another thing we're going to explore in our lab on Tuesday. Glycosidases are enzymes that catalyze the hydrolysis of various types of glycosidic linkages. They break these glycosidic linkages. Specific glycosidases are needed to hydrolyze these different bonds. We will look at examples in class on Monday. So polysaccharides are biological polymers. They have glucose monomers are the most common. They can be alpha 1 to 4 linkages or beta 1 to 4 linkages. So polymers that are any large molecule with the same or similar repeating structures. And that's really all of our macromolecules that we're going to talk about, whether it be carbohydrates, lipids, DNA, or proteins. But specifically here, we're talking about chains of monosaccharides making up polysaccharides. So the most important ones, but certainly not the only ones, are amylose, amylopectin, cellulose, and glycogen. We're going to look at the structure of these. Let's start with starch. So starch is amylose in part. 20% of starch is amylose, and that's an alpha helix. Remember, our DNA is an alpha helix shape. It's the winding shape. Amylopectin, on the other hand, is 80% of starch, and it is branched, kind of looks like a tree branch. Plants synthesize this starch, and they also synthesize cellulose from glucose. They're just different configurations of glucose. Kind of like carbon is very different when configured in different ways. For example, pure carbon graphite slides across each other and is brittle, while networked carbon in diamonds is considered the hardest substance on the planet. But plants use starch to store energy. 20% of starch is amylose, 80% is amylopectin. But all are made up of glucose. So here's the helical shape of amylose, and here are the linkages. They're alpha 1,4 linkages, repeating over and over. So alpha means that these glycosidic bond oxygens face down. So you will see, and you can see, this is glucose is a hexagonal sugar. So these are the glucose molecules, and they make these repeating shapes based on how the glucoses interact with each other. Amylopectin is branched, and instead of 1,4 linkages, it has alpha 1,6 linkages. But it has almost like a tuning forks attached to each other type of configuration, whereas glycogen is very branched and kind of very much looks like a tree. Cellulose is unbranched, and it's an opposite of amylose. It's a beta 1,4 glycosidic linkage, and you can see the effect is is very different. It's flat sheets that are parallel instead of the helical. So this is what cellulose fibers look like under the electron micrograph, and they are very important to the structural aspect of plants. They give it the strength to hold up giant mass in some cases. If you think of a sequoia or oak tree, the cellulose in its trunk holds an incredibly heavy load, not just from the branches, but from stabilizing the roots beneath. Glycogen is the storage form of glucose. When you get insulin high levels in your blood, your body triggers glucose to leave your blood system, go to your liver, and be stored as glycogen that can be reversed and used as energy later on when needed. In plants, they store their energy in the form of glucose. You can see this in the orange-red micrograph is the starch. So when we consume starch, we hydrolyze it, we break it down, we break those glycosidic bonds, and it becomes glucose. So then glucose further breaks down into CO2 and H2O, and energy inside the cell via several step biochemical processes. Excess blood glucose is converted into glycogen. That's the polymeric storage form of glucose for mammals. So you can see glucose is what our food is broken down into. It moves to our bloodstream, takes oxygen for the cellular respiration to happen, and we get energy in the form of ATP and H2O and carbon dioxide. So amylose, amylopectin, and glycogen are primarily joined at the alpha 1 to 4 linkages, but there can be a varying amount of glycosidic linkages in the polymer, about 250 to 4,000. Amylose is a minor constituent of starch. It's unbranched, but it has a helical shape, so it's different distinctly from the other ones in this case. So we're going to do a test for amylose. It binds to iodine, and iodine has that distinct like blue-purple color. So when we test for starch, the iodine can be used as an indicator to see if starch is present because of the amylose content of starch. Amylopectin is a major constituent of starch. It's branched, kind of looks like tuning forks linked together. It's an alpha 1,6 linkage. So oxygen pointing down between carbon 1 and carbon 6 of adjoining glucose molecules, and altogether it has like a spherical shape. Glycogen is highly branched, very similar to amylopectin, except that it's alpha 1,6 branches, and they occur every 10 monomers, making it more branched than amylopectin, which is more like 30 monomers. Glycogen is how glucose is stored in mammals. Cellulose is unbranched. It's the main component of wood, paper, and cotton. It serves a structural role in plants that we talked about, allowing strength and ability to stand for plants, even in like high rain and winds and erosion of soil. So this rigidity of structure comes from extensive hydrogen bonding between the sheets of the polysaccharide, so they're hard to break apart. So alpha glycosidases are able to break down starch, but not cellulose. Humans do not have beta glycosidases, so we cannot digest cellulose. And ruminant animals have a lot of help from their microbiomes to do that. So they've got bacteria in their gut that produce beta glycosidases, so they're able to use grass as fuel. But you can see this guy is mid-cud, so he has 4 parts to his stomach, and one is the rumen or fermentation vat. So he eats grass, or she eats grass, and it's digested once, regurgitated into the cud, you call it. That's what ruminant animals means. They chew cuds, and then they get a second round to digest. This is what a cellulose crystalline subject looks like. You can see very tiny within the plant, but repeated over and over again. So oligosaccharides are cell markers that people receive organ transplants must take immunosuppressant drugs for the rest of their lives so they won't reject the transplanted organ. These oligosaccharides are sticking out of your cell membrane, and they're like the chemical detectors. If they find foreign substances, they attack it and destroy it. And so that's what your body would do with a transplanted organ to protect itself. Cells contain chemical markers on their surface. This is an oligosaccharide that would be a sensory for foreign hosts. Most oligosaccharides are covalently bonded to lipids and proteins in the cell membrane, and they can be completely embedded or partially embedded in the cell membrane, so they stick out into the extracellular matrix outside the cell. So 4 blood types, AB, and O, and of course we have the positive and negative in there. They differ in markers on their cells. So type O has a trisaccharide marker. So type A has a tetrasaccharide that includes 3 monosaccharides from type O. Type B has different tetrasaccharide that includes 3 monosaccharides from type O, and AB combines the markers of A and B. So there are multiple alleles that go into that. So people with blood type O are universal donors because they have a trisaccharide, and A and B includes its 3 monosaccharides. People with type O can accept the blood type O only. All other blood types have foreign markers, so they're not recognized. So there's universal donors, but AB blood is a universal recipient because they have markers that are common to AB and O. And that is the end of chapter 11.